So for birds,
That is the objective section.
under the objectives, yeah. So it says, recognize species, birds to order. Okay, so you need to know everything by order, by their formal name. You need to know that. Recall the orders discussed and their distinguishing characteristics, and then classify any skin or photo to the order. So that's the first level, you need to know the order. Then you need to recall, the order, recall the subfamilies of Anatidae and those in BC, distinguish among the perching ducks, dabbling ducks, flapping ducks, and then recall the life histories of BC red-listed species, and it's here. Somewhere it says it's by the sheets provided, but recall the names of selected red-listed species, recall their biology, which is distribution, key habitat features, and important life history traits. And then in the brackets, it says migration, aquatic, and then see the attached data sheet. So those are the 10 sheets that you need to look at. So of all the birds, we only need to know the red listed ones on the data sheets. Yep. Okay. And then recall where to get the data. And the E, you already know E4, or sorry, GC, System Explorer. Recall definitions as you had on your final as the midterm exams, such as the COSEWIC and the provincial ones. And then distinguish between the following birds: common raven versus crow, sharp-shinned hawk versus spook versus three grouse species, for example, grouse out. Mountain versus black-capped chickadee, downy versus peregrine. So that's. And then there's the introductory sections, which are lecture exams, which you've already had. And then the bird calls.
So Doug, if we're given one of these birds on the lab exam that isn't red listed and that we aren't trying to distinguish between,
Yep.
we just have to know the order.
That's right.
Okay.
Or subfamily,
Or subfamily for the water.
Oh yeah, yeah.
Okay. Okay. For the amphibians and reptiles, recall orders discussed and the families discussed and their distinguishing characteristics. Okay, so as different than birds. Bird is order versus the subfamilies or families for the waterfowl only. The amphibians and reptiles and amphibians, you need to know all the orders and the families. Recall the families discussed and their distinguishing characteristics. Classify any skin or photo to family or order. That's consistent with birds, except the emphasis on birds is order with the various subfamilies. And then the life history traits. Recall which species are red listed. For those, this handout is the updated one. Okay, in that all the labels that are red labels and blue labels have now some of the status has changed. So don't follow the color of the labels. We'll have to redo those. But in here, the handout says which ones are red and blue listed, and then there's a table at the back that says which ones are red and blue listed. Okay, so these are the ones you need to know. Okay. Distribution for those guys, most of them should have the distributions on the back, and if they don't, then you would use your book. Okay. Pretty well all of them have these cards, but a few don't, and I will try to find out where those cards went to. But in the ones that are missing these distributions, then the book would be the one that you would go to. When I do my maps on the lab exam, I generally pull out of the book. Okay? Because some of these maps are like 40 years old, but they will closely represent exactly what's there. Okay? It's going to be very obvious. I'm not going to have something where one of these might extend up to Merritt and the other one extends up to Kelowna. Not getting that. It's going to be coastal, northern, eastern type thing. It's going to be very obvious where you can look for these guys, where you can't confuse these guys. Okay. And then small mammals. I have to look it up because I printed it, but I forgot to snag it from the computer. Small mammals. Family and order, or order and family, same as the amphibians and herps. Recall the distinguishing characteristics of the orders and of the families and subfamilies. Basically, the subfamilies would be the old world mice versus the new world mice versus the voles and lemmings. Okay. Recall the life history traits of select red listed species, and they are, see the attached data sheets. And you get same thing, pretty well same. Distribution and key habitat features. Okay, so where would you find the red listed snowshoe hare species? Any idea?
Sorry?
Where would you find the red listed subspecies of snowshoe hare?
Fraser Valley.
Yeah, the lower Mina. So it's just things like that. So when you're out there, and the reason we do this is because multiple people on their interviews literally have been asked, list 10 red listed species you might find in this park. And it's part of the job that you need to know where these guys are. So then we've added this in where you're getting a select number of red listed species to understand it. It's, goshawks, massive money going into doing surveys for goshawks. So you should probably know where goshawks are.
Sorry, where are we getting these updated from off of the ecosystem orders? And also,
From the, well, these guys have the maps associated with it. If you turn those cards over, there's a map on, oh, actually on the very front.
but the status is what I'm saying. Or
So
where
the
we just do that.
status are of the 10 sheets that are for the birds, 10 species only, or if it's 10 or 9, on the special data sheets that look like this. Like these guys, okay? Small mammals, 10 data sheets. These are the ones you need to know red listed species from, okay? And both the birds and small mammal cards like this are on the line, or on the course outline, okay? The difference is amphibians need to know all the red listed species. Birds, just to select 10, and then, or I'm going to guess 10, I've taken some out. I can't remember if I started with 12 or 10, but the data sheets are online, and for the small animals, the data sheets are online. But for the amphibians, there's no data sheets, because you just need to know all the red listed. But the red listed status is in red notes, with all the red listed status. Does that make sense? I mention that now just because I'm going to be asked about 15 times in the course of the list. And my answer will always be, what does it say on the front page of your handbook? Sometimes it changes from year to year. So then I always hold my guess.
And so there's also going to be questions that are not on the first page that are listed as lab.
But the lab exam, yes, because the lab exam is 100%. What family does this species belong to? What order does this species belong to? What is the distinguishing two key cranial distinguishing characteristics? That's what the lab exam is. Okay, so anything outside of that is not conducive to 90 seconds, a minute and a half of answer. Okay, so it's all identified as a species. If it's a red listed species, it could be what map best represents the distribution of this species. And if it's one of those 10 on the sheet, then you'll have 3 maps X, Y, Z, and you just have to fix it. Make sense? So that's the key. There's no short answer, like no essay questions, obviously, in the lab exam. They're not long questions. It's just 1 and 2 and 3 word answers.
How many specimens will be on the model?
Usually there's 30 stations and 2 questions per station. So question A could be a mountain beaver. Question B could be side, it could be a different species. So then it could be 60 questions, up to 60 questions. Sometimes, most often, question A is identify this critter as a family. What is the key distinction? And then question B is what is the key distinction characteristics of this family? So normally one's a follow-up, but not necessarily. One could be name the family, name the A, name the B, completely different order. So I hope you guys had lots of lab exam questions, so I think, or with applied ecology and plant course and stuff. So I think you're probably reasonably familiar with the lab exam.
Have you done this in bird calls?
That,
You have a recording of the bird calls?
yeah, I'm gonna play them through the speakers up there.
Oh, okay.
And everyone's gonna stay at their station. And then I'm gonna say, this is usually, it's questions 25 to 30. I'll say 25A, play the call twice, and then give you usually a minute to answer. Then here's bird call for 25B. Play that call, you write down what you think.
And it'll just be like, what is the species during the bird call? And we'll be like,
No, what is the species? Yep.
Common.
Common name, yep. No, any of the formal names are gonna be the handwritten stuff. Okay. That help? That make sense?
So for the yellow and listed species.
So you only need to know red listed
Yeah.
for amphibians and birds.
Okay.
Or herpetiles, amphibians and reptiles.
So for the yellow and blue listed that are out, we need to know like what they are on the picture, or are we gonna?
I can put a picture of any and herp out, and you need to tell me what family, what order it belongs to.
Okay.
So that's why all the pictures are out, because I could pick something like, well, you have to know what a Russian redback is, those amphibian ones. You need to know Enstina's Western redbacks, but I could put out any of these guys, independent if they're red listed or blue listed or yellow listed, and you should be able to put it into family, based on the features.
So not necessarily the exact species, but the family.
Yeah,
Okay.
there'll be a random collection of specimens that belong to the family, and a specimen will come out and it'll be what does this family, or what is the order, or what is the distinct characteristics, or what map best represents the distribution of.
Okay.
If I'm asking about what red listed, what map, you automatically know I'm either on the sheets for the small animals, the sheets for the birds, or any red listed guy here. Okay. I may not even ask what the species is to say what distribution best represents for this specimen, and then you just have to pick the one. So most of them have, like say, the map. Some of them may not, but you'll have to go to your books. But most of these guys have distributions on the cards in front of you. Okay.
Thank you.
Hopefully that's, in the actual lab, it doesn't look too bad. Most time in the lab, most folks were done in like 20 to 40 minutes. But now when you see it all out, it probably looks a little overwhelming.
You guys
I think these.
We don't need to know.
What? Japan listing, I know. I would take pictures of it. Let me send me these. Send me these pictures. Share it in the chat. Nice.
Yeah, that's
Yeah, or like, yeah.
but like I just seen
Yeah. Minutes for each stuff.
But if you're only asking like family.
5 minutes discuss.
Gary. So you get like a minute and 20 seconds, like, or 15. Alright. We'll get there.